Hello everyone. Welcome to Progressive Education Society's Modern College of Pharmacy, Nigdi, Pune. Today we will be discussing HPLC. HPLC stands for High Performance Liquid Chromatography or sometimes referred as High Pressure Liquid Chromatography. This technique is essential tool in both biochemistry and analytical chemistry for separating, identifying and quantifying components in a mixture. HPLC offer high speed and resolution compared to traditional chromatography because the mobile phase is pumped through the system at high pressure. What is chromatography? Chromatography is a method of separating components in a mixture between two phases, a stationary phase and a mobile phase. The stationary phase, which can be solid or gel, adsorbs the components, and the mobile phase, which can be liquid or gas, carries the analyte through the stationary phase. Principle. The principle involved in HPLC can either adsorption or partition. Interaction of sample with both stationary and mobile phase. The analytes are injected into the flow of mobile phase. The outlet of the column is connected to a detector where the elute substance are detected. The separation is achieved in the column packed with stationary phase material of low particle size. The reliable flow rate of the mobile phase with appropriate pressure is applied and the sample mixture is interact between the stationary phase and mobile phase. The interaction between these phases result in separation. Classification. HPLC can classified into two major types: normal phase chromatography and reverse phase chromatography. In normal phase chromatography, stationary phase is polar hydrophilic and mobile phase is nonpolar hydrophobic. In reverse phase chromatography, stationary phase is nonpolar hydrophobic and mobile phase is polar hydrophilic. Polar bonds and nonpolar bonds have more affinity than polar nonpolar bonds. Reverse phase chromatography is more commonly used as drugs are usually hydrophilic. The elution in HPLC can classified into two categories: isocratic elution and gradient elution. The isocratic elution, in this technique the mobile phase composition is fixed constant throughout the chromatographic procedure. For example, if a method consisting of mobile phase as methanol and water in a ratio of 70:30, the same ratio is maintained for the entire chromatographic procedure in isocratic method. In gradient method, the composition of mobile phase is changed either stepwise or continuously as elution proceed. For example, initially a composition methanol and water 70:30 for some time period, let's say 10 minutes, is maintained. Then the polarity is modified by changing the ratio to 80:20 for next 5 minutes and then to 90:10 for another 5 minutes. Changing the composition to bring out the desirable separation is called as gradient elution method. Next, we will be see the instrumentation part. The instrumentation consisting of the mobile phase reservoir, pumping system, sample injection system, column, detector, and data collection device or recorder. Here is the visual representation of the instrumentation of HPLC. Mobile phase reservoir. Generally, reservoir made up of glass are used. One or two reservoir of 500 ml capacity or more quantity if required are used. The quantity of the mobile phase to be used in the separation depends upon the nature of solvent, quantity of mixture to be separated, time required for the separation, particle size of the solvent, type of stationary phase used and another such factors. Next is the degassing. It is necessary to remove the dissolved gases present in the mobile phase solvent. Different techniques are followed for degassing, which are discussed below. First is external vacuum degassing. In this method, the solvent in a container is kept in a ultrasonic bath. Ultrasonication is done under vacuum using a vacuum pump. Here is the visual representation of external vacuum degassing. This process will remove the dissolved gas from the solvents. Next is the helium. In this technique, helium is bubbled into the solvent, which will remove the other dissolved gases. Helium is insoluble in the mobile phase, so it escapes out without interfering. This can be done online or offline. Next is a pumping system. The mobile phase is forced from reservoir to the column using pumps more resistant to the flow of mobile phase due to the small internal diameter of the column and low particle size of the stationary phase. Hence, high pressure is required to push the mobile phase with uniform flow rate. This is achieved by the pumps. The general requirements are it should provide the desirable pressure, generally up to 6000 psi. It should provide uniform flow rate ranging from 0.1 ml to 10 ml per minute. In case of preparative analysis, it should provide more flow rate. It should provide pulseless flow and chemically resistant to all solvents. It should provide reproducibility. Syringe type pump consists of large syringe-like chamber suitable for small bore column, and these pumps have a volume between 250 to 500 ml. Double syringe pumps have also developed, in which one piston is delivering the solvent to the column while the other one is refilled from reservoir. Here is the visual representation of the syringe type pump. Next is a reciprocating pump. The term reciprocating describes any continuously repeated backwards and forward motion. This pump is widely used in reciprocating pumps. Basically, the movement of the plunger piston causes suction of the eluent during the backward stroke and the delivery of the pressurized solvent during the forward. Constant pressure pump. In this type of pump, the mobile phase is driven through the column with the use of pressure from the gas cylinder. A low pressure gas source is needed to generate high liquid pressures. The valving arrangement allows the rapid refill of the solvent chamber, whose capacity is about 70 ml. This provides continuous phase flow rate.
Sample injection system. The injection is located on the high pressure side of the pump. Sample injection valves allow effective sample introduction without interrupting the flow or altering the system pressure. Components of the sample injection valve: a needle or a syringe to pierce the vial septum, a metering device to measure the aspirated amount of a sample liquid, a loop or a holding device to retain the sample prior to injection, a valve which is used to alter the hydraulic path of the eluent through the device in order to effect direct injection of the sample plug into the eluent stream under pressure. Columns. The requirements for an ideal HPLC columns are as follows. It should have a uniform column packing. It should have a spherical particles. The particles diameter should be ranging from 3 to 10 micrometer. The porosity of the particles should be in the range of 50 to 70%, extending to 80% for the size exclusion chromatography. The column should withstand the pressure during operation. It should give reproducible results. It should be easy to handle. The particles should not shrink or swell with the nature of the eluent. It should have a uniform particle pore size distribution. Types of columns. First, guard column. Guard column is the column placed before the analytical column. These columns also called as pre-columns. They are used to protect the analytical column from the impurities and other contaminants from the solvent. It also removes the components that bind irreversibly to the stationary phase. The guard columns are compulsorily used during the bioanalytical studies to protect the analytical columns from biological matrix. Second, analytical column. Analytical column is considered as the heart of an HPLC system. The reason is that this is the part where the separation of the mixture takes place. The efficiency of the separation is purely depends on the columns. Types of analytical columns. First, small bore columns. Columns having the diameter less than about 2 mm. They are also known as the micro columns, that they are operated at much lower mobile phase flow rates. Second, 3x3 column short. Columns packed with 3 micrometer bonded silica stationary phases have a sufficient efficiency for many separations. Third, monolithic columns. It has a pore channels rather than being packed with beds. Their construction is more similar to rod with a random channeling. Detectors. Detector is a device which detects the eluated compounds emerging from a column. An ideal HPLC detector should meet the following requirements. It should respond to wide variety of components. It should be compatible for the temperature and flow rate changes. The sensitivity should be high. It should have a low dead volume. It should give at least selective response and specific is preferable. It should give reproducible results. It should be compatible with a liquid flow. Types of detectors. Bulk property detectors contains the refractive index detectors. In a solute property detectors, it contains the UV visible absorption detector, fluorescence detector and amperometric detectors. Others detectors, infrared absorption detectors, evaporative light scattering detectors and mass spectrometric detectors. Bulk property detectors. Refractive index detectors. Detection occurs when the light is bent due to sample eluting from the columns, and this is read as a disparity between the two channels. It is not much used for analytical applications because of low sensitivity and specificity. When a solute is in the sample compartment, refractive index changes will shift the light beam from the detector. Solute property detectors. UV visible absorption detectors as seen in an image. UV visible detectors. UV visible detector is widely used as it detects large number of compounds because of most drugs have appropriate structural characteristics for a light absorption. These are useful for aromatic compounds and other type of unsaturated systems. These are classified as fixed or variable wavelength detectors. Fixed wavelength detectors employ filter as a source to provide appropriate wavelength. Most common fixed wavelength detectors are based on a 254 nanometers. Variable wavelength detectors are employed a spectrophotometer to provide dispersion of a light and selection of any wavelength in a UV visible region. Diffraction grating are frequently used for a wavelength dispersion.
Photodiode array. A photodiode array is a linear array of discrete photodiodes on an integrated circuit chip, that is called IC. Second, array detectors are especially useful for recording the full UV visible is a absorption spectra of sample that are rapidly passing through a sample flow cell, such as in an HPLC detector. Next, fluorescence detectors. It is based on the fluorescent radiation emitted by some compounds. Second, the excitation source passes through the flow cell to a photodetector while a monochromator measures the emission wavelength, more sensitive and specific. Third, the disadvantage is that most compounds are not fluorescent in nature. Amphometric detector. A amphometric detector works based on the reducing and oxidizing property of a sample when a potential is applied. Second, the diffusion current recorded is directly proportional to concentration of compound recorded. Third, advantage, highly sensitive detector. Disadvantage, this detector is applicable only when the functional group present in the sample can be either oxidized or reduced. Next, data collection device or recorder. Recorders are used to record the responses obtained from detector after amplification. Second, they record the baseline and all the peaks obtained with respect to time, but the area of the individual peaks cannot be known. Third, integrators, improved version of recorder with data processing capabilities. Fourth, the height and width of peaks percentage area, integrators provide more information on peaks than recorder. Applications. Pharmaceutical industries. It is used in departments such as R&D, quality control, F&D, assay of pharmaceutical drugs, multi-component analysis, analysis of polyphenol, steroid, vitamins. Second, stability. Studies stability of product, degradation studies such as acid-base oxidation, photodegradation. Bioanalysis. India to Thailand used to determine the drug in biological metrics such as blood from plasma, urine, serum, feces. Fourth, natural product analysis. Standardization of herbal extract. Second, standardized on polyherbal formulation. Fifth, food analysis. Analysis of honey sample. Second, food formulation. Third, dairy products. Fourth, sugar analysis. Fifth, analysis of nutrition value in nutraceuticals. Sixth, drug interaction studies. Useful in drug interaction studies.